If it does not have CD16, that means it's a classical monocyte, because CD16, just another cell marker, is an indicator of an activated or an inflammatory monocyte. Then we have that high inflammatory group that has double pluses there, really high expression of CD16. So that's how we determined which monocytes are which using flow cytometry. And here are a couple of charts to show you how it works. This is our blood sample. This is what we see from the flow cytometer. This is debris and junk. These are all of our granulocytes, mainly neutrophils, some probably eosinophils and basophils out here. These are lymphocytes, and these are our monocytes. So R1, we drew this circle, which is called gating. We wanted to look just at the monocytes. We are going to analyze all these other cells, by the way, but obviously we don't have time to talk about all that today. In any case, we can gate on the monocytes and pull it over here and ask it how many have CD14 and how many have CD16. These cells here would be CD14 negative. These cells here would be CD14 positive. So we have two populations. These are our true monocytes. That's some other junk that might have gotten caught in that gate. These are CD16 positive. These are CD16 negative. So using that, we can determine these are our classical monocytes, our low inflammatory, and our high. And you think, we thought, at first I thought we're not differentiating between them. This looks like one big population, but we actually did some controls to determine where to put this cutoff. So really they bleed in among one another, the low and the low inflammatory and the classical monocytes. So just to give you an idea, so these are, each dot is an actual cell and it plots it so we can determine what we want to know. So here's some summary. Without taking into consider groups, we looked at the difference between the high, the low, and the classical monocytes in regard to MC1 and MC3 expression. This is shown in mean fluorescence intensity, and what that means, these are actually the markers, or the cell surface markers, are tagged in flow cytometry with a fluorescent marker. You stick an antibody with a fluorescent marker on there. It binds to your receptors, your CD14, 16, whatever. And when it traverses through the flow cytometer, or the cells are excited with a laser, and such that those fluorescent markers fluoresce. So that's how we get all of these cells to show up. So the mean fluorescence intensity, how much fluorescence was there, is directly proportional to how much MC1 or MC3 is there. So here's what we saw. Those cells that were high inflammatory, the CD16 positive, had significantly more both MC1 and MC3 receptor. And that makes total physiological sense, because they are activated, they need that control, the anti-inflammatory function there to help control inflammatory processes, and that they've been activated. They're a little bit more in low and classical, but these were not significantly different from one another, but we'll see. We've not finished this study, so once we gain all of our subjects, who knows what we'll find. So no one has ever reported this. We were pretty excited. It fits what we might have assumed would occur. Now, it'll be really interesting to find out, again, the genetic expression data and were these actually producing more or less cytokine from the culture wells and so on. This graph includes the MC1, MC3 on high, low, and classical monocytes in our different groups. So far in lean fit we had 14 subjects, that's the dark purple. Obese fit, we had 8. Lean sedentary, we had only 5. Those are all women. We need men. So if you know anybody who just doesn't exercise regularly but is normal weight, they don't have to be lean, send them my way. And then finally, these are our obese sedentary, 14. So what we see here, there was no statistical difference among them yet, but notice this huge variability in our lean fit folks. Compare that to the variability in the other conditions or other groups. There's something going on with fitness. We just don't know what it is yet, and again, we'll have to wait to finish data collection. But we're speculating, are there responders and non-responders? Everybody's familiar with that particular effect. So we just don't know yet, but there's something maybe happening. So we'll be looking forward to finding out exactly what that is when we finish data collection. So the significance of this current study, and hopefully future studies, is that it will help us to understand how exercise is good for you. And due just to the anti-inflammation effects of melanocortin, clinical trials are ongoing now to determine if alpha-melanocortin stimulating hormone may be used as a medication, a drug, to reduce some of this inflammation. So if we can show that exercise does it instead, again, we have an idea for you other than taking a pill. I don't know how well that works for everybody, but and of course we'll understand potential treatments. This accompanies what I just said. And we are the first to examine the influence of fitness or obesity on peripheral MC receptor, or excuse me, not obesity, fitness and its interaction with obesity on MC receptor expression in humans. Very little work done in humans. And I'm working now with a colleague at Louisiana State. She's the one that published some manuscripts where resistance training and aerobic training reduce CRP. I told her about some of our data, and she's real intrigued. So we're going to try, based on all the data we have now, I think we have enough preliminary data for a pretty good NIH grant, and we'll probably collaborate with Dennis Cheek and Mike Chung and a few others here. So I would like to do that in the next probably year or so, maybe a year and a half. You know how that goes. So that is where I am, and I stole this slide from Dennis Cheek a long time ago, and I always like to show it. So thanks for your patience and for listening, and if you have any other questions, I'll answer. If not, I'll see you later.